Stay safe.
Will do.
All right, so small class today. Only a handful of people on board. We'll wait another minute. Everybody hear me okay? See the screen? Thank you, thank you. All right. So we'll go through those quiz results in a moment. I think everybody did pretty well. And hopefully the format was okay. I'm not as experienced doing those quizzes as Dr. Octung, but hopefully it worked okay. And so then the problem set is posted. I don't know if anybody's looked at that, but make sure you look at the instructions. We will go through how to solve that problem starting in about 15 minutes. Okay, and then we'll also cover that on Wednesday. So you should be able to do that, but maybe some of you have already gotten a start. I don't know how many of you have bought the book or have a book, but there's probably information in the chapter in that book that will also help you. Okay, so let's take a quick look at the quiz questions. And so if you have any questions, I think the answers, again, I'm not sure how when you took the quiz, if it showed you the right answer, it showed you if you got it right or not, showed the right answer. How'd that go? Did I have it set up right? Did it show you the?
It didn't show us anything.
I'm sorry, Emily, say that again.
It didn't show us anything after the quiz. It said the quiz was muted.
How did Octung have it set up?
She had it set up so that we knew our score right away, and it showed us what we got wrong.
Okay, so I need to ask her how to make that happen. Sorry about that. So how did you know what you got? Just by in the grade book?
Yeah, once you released the grades, then we saw it.
Okay, I apologize. I'll set that up differently. I had her check through everything, but I think she was mainly looking to make sure I had the other part, the actual questions and not the grading, all the right features. So I apologize for that. Hopefully you weren't waiting too long to see what happened. So I think the way I copied it over into here, all the answers are A. So hopefully they weren't that way when you took it. It's supposed to have randomized the answers. The microbe that has the lowest water activity requirement is Staph aureus. The antimicrobial used in wine, and that's antimycotic, was sulfite. MA in meat is to control cyclotropes like Pseudomonas. The application for this beverage, pH 3.8, would have been benzoate. Aseptically processed foods use H2O2, hydrogen peroxide, to manage for the packaging. The generation time was 2 hours. Nitrite is to inhibit germination of Clostridia, and the reason why benzoate and not benzoic acid was used was because of the solubility thing. We went through that pretty carefully. All right, so that's, if you have questions about that, let me know. And then just quick review. Again, we just talked about benzoic acid. It was on the, it's an important part, it was on the quiz, and we talked about how it's active, the mode of action, and this could be benzoic acid, but it's the same if it were sorbate or propionate. They all have the same mode of action. They acidify the intracellular, the cytoplasm, and in response, the microbes want to pump out those protons. So they spend their ATP on that, and eventually they run out of ATP, and the inside becomes acidic. We could add sodium or potassium salts of benzoic acid, sorbic acid, propionic acid, in that case, probably calcium. And those acids push the salt to the acid form. All right. And then we talked about acidulants, lactic acid and acetic acid. They are mainly used to lower the pH. They do have the same mode of action as those weak organic acids, but most of the time these are added to acidify the medium. Okay, and then we talked briefly, very briefly, about natural preservative agents. These are there for usually other things. Herbs and spices are there for flavor, but they can be inhibitory. One of the downsides for using them in applications is that they do contribute flavor. So it's hard to remove the to separate the antimicrobial from the flavor. Smoke is very effective. You could smoke naturally and produce all sorts of antimicrobials, or you could actually condense the smoke and make liquid smoke, and that's a very common food ingredient then, mainly for flavor, but it does add antimicrobial properties. And then we talked again briefly about lysozyme from egg white, lysis bacteria, used in some countries, not much in the US. Same for lactoferrin, binds iron, keeps the iron away from the microbes that want the iron. And again, limited applications. And same thing for lactoperoxidase. It's found naturally in milk. It catalyzes this reaction that leads to the formation of this thing called hypothiocyanate, which is an antimicrobial, but it's difficult to kind of monetize or commercialize this whole reaction. Okay, and then I did briefly mention these antimicrobial agents produced by microbes, biologically based antimicrobials, which were two main types, antibiotics and bacteriocids. And in the interest of moving ahead to get to the next section, I'll skip that and we'll come back to that next week. Okay, and then the fifth type of antimicrobial agent, so there was inorganic, there was organic, there was, let's see, what was the third one? Was natural, and the fourth one was the biologically based, and then the fifth one is indirect. And these kind of are similar to some of the naturals. These are substances added to food for other functions, and maybe you could put the herbs and spices in here, I suppose. But these are things like menthol, vanilla, diacetyl. Diacetyl is butter flavor. You could even include some of the antioxidants. And again, I mentioned the spices and herbs. So these are there for other functions, but they have antimicrobial activities. Okay, so now let's recap how to inhibit growth of foodborne organisms. Temperature, water activity, EH, atmosphere, antimicrobial inhibitors, and lastly is pH. And we're just going to spend a few minutes on this because we've actually talked a little bit about pH when we talked about organic acid preservatives. So you can classify foods according to their pH range, and you'll see this four categories or classifications: high acid, acid, medium acid, low acid. But I prefer just putting a line right here at 4.6 and just referring to these as acid foods and non-acid foods. Okay, so I don't make too many distinctions, where the cutoff is 4.6. Now, so and the 4.6 is important because pathogens won't grow below 4.6. They can conceivably grow above 4.6, but in the larger scheme of things, spoilage microbes, there are microbes that can grow below 4.6. So this 4.6 is a pathogen cutoff, and the main pathogen we're concerned about is CBOT. Okay. So most pathogens cannot grow in the acid range, but that's not to say that there aren't spoilage organisms. And we know this because you will have, you'll see microbes grow in your ketchup, or, for example, that's acidic, and you'll see mold grow on there occasionally, and other acid foods. Okay, so foods below 4.6 are generally considered safe, but not immune from spoilage. Okay. Now foods, and we'll talk more about spoilage in the last section of the semester. Foods not naturally acidic, so things that are naturally acidic would be fruit juice, fermented foods, and so forth, can be made acidic by adding acidulants. And so remember, we just talked about lactic and acetic can be added to things. So when people make homemade pickles, they take cucumbers, which have a more of a near neutral pH, and they add vinegar. They could add salt brines, but typically they'll add some vinegar. Phosphoric acid, citric acid, these are acidulants. Their intent is to lower the pH, generally to below 4.6. Okay. They may have weak organic acid functions, like we just said, but their main function is to lower the pH. Okay. And then, as I just mentioned, aciduric, acid-tolerant organisms are common in foods and can still spoil acid foods. So these would be lactic acid bacteria, acetic acid bacteria. Obviously, if they produce lactic, they're going to be tolerant to lactic. If they produce acetic, they'll be tolerant to acetic. And then yeast and molds are definitely problematic. Okay, so that's all we're going to say about pH, but we'll come back to that as we talk about spoilage, and even when we talk about killing microbes, which is our next topic. So we've covered keep out rule, can't emphasize that enough. Remove, we just spent a week on prevent delay growth, and now we're going to spend a couple lectures on the next one, kill and destroy, killing microbes. And food microbiologists love to kill microbes because that provides a much better level of confidence that we preserve the food versus some of these other ones, which have some, they're not perfect. So we can't keep everything out. We can't remove all of them. And whenever we try to prevent delay growth, we're always selecting for somebody else. We like to kill. Okay. And we're going to mainly talk about temperature. We'll talk a little bit about irradiation. We'll talk a little bit about high pressure, and we'll briefly mention some of the other ways, but our focus is on temperature. Okay, so this next lecture from here on in for the rest of today and into Wednesday, we're going to cover some really important concepts that are key to this entire semester. So I know that Dr. Aucton would probably say that, understanding pathogenesis and virulence is the crux of the class. I would say that understanding some of these concepts for how food microbiologists inactivate microbes is one of the central points of this course. So really important concepts, especially if you work in the food industry as a microbiologist or processing. Okay, so we'll start by defining a couple of key concepts. We'll start with pasteurization. So sometimes when we think about killing microbes with temperature, we're thinking of canned foods, but there's lots of ways that in food science that we use temperature. So one is pasteurization, and the other one is, like canned food, sterilization. Pasteurization, the purpose is kill, and I've chosen my words carefully here, kill vegetative pathogens capable of growth in the food. Okay, so this could be milk, but it doesn't have to be milk. We pasteurize all sorts of things. We pasteurize beer, we pasteurize fruit juice, but milk is the best example. Vegetative pathogens capable of growth in the food. Another goal of pasteurization is to reduce the number of spoilage organisms in the food. So this is different from pathogens. We want to reduce spoilage organisms in food. Again, milk is a good example. Okay. Even if we collected raw milk with no pathogens, we'd still want to pasteurize it to reduce the number of spoilage organisms so that we can get shelf life. So we can get 3 weeks perhaps, or at least 2, maybe 17 days of shelf life. Okay. And a side benefit of applying heat is that we can inactivate enzymes involved in spoilage. Remember, we talked about proteases and lipases that could be produced by microbes. Some of them are heat stable, but some of them we can inactivate. Okay. The purpose of sterilization is to kill all pathogens and their spores. So here we're just killing vegetative. Here we're killing all pathogens and their spores. So when we pasteurize milk, if there are C-bot spores, as it's possible, there could be C-bot spores in raw milk. Pasteurization does not kill them. So is that a problem? Why is pasteurized milk safe from Clostridium botulinum? And that's not a rhetorical question. You can answer that. That's a really good question, and I think it deserves an answer.
So I think it depends on how the milk would get stored after pasteurization. It's in aerobic condition or in non-anaerobic condition.
Okay, and how is milk stored?
It's stored in aerobic condition.
Exactly. So Antar did a good job on that. So pasteurization says kill vegetative pathogens capable of growth in the food. So even if C-bot was in the vegetative form, it's not capable of growth in the food. The milk is too aerobic. That EH of milk is high. There's lots of oxygen in the milk. In fact, milk can spoil by oxidative rancidity if we're not careful. So those pathogens won't grow, C-bot won't grow in normally held milk. Okay, let's continue. Sterilization, kill all pathogens and their spores, all organisms, now I'm not even distinguishing pathogens, capable of growth under normal conditions. So even vegetative cells, even, lactic acid bacteria, capable of growth under normal conditions. So it's killing everything in there, just about. But here it says, food sterilization processes do not make the food clinically sterile. I'm not sure even what that means. We'll have to come back to that. I do know what it means. Okay, so we're going to talk a little bit more about the sterilization and this concept of clinical sterility. Food sterilization do not make the food clinically sterile. Okay, so let's say you had to go to see the dentist, and you get in the chair, and or perhaps you work in a vaccine lab or a pharmaceutical lab. But let's go back to this dental chair. Wouldn't you want to make sure that those utensils go into your mouth are clinically sterile? You don't want any chance that there's a microbe on those devices. Okay, so in an operating room, in your mouth, in a pharmaceutical lab, or in a production facility, you want clinical sterility. Okay, you don't want any chance that there could be a microbe. Okay, so that's as close to absolute sterility as you can get. Okay, in food processing, clinical sterility is not required. So these cans are not clinically sterile. This aseptically packaged soup is not clinically sterile, and this pouch is not clinically sterile. Why don't they make it clinically sterile?
Because that's very difficult.
Okay, you could do it, but why not? You could do it. Why
Isn't
not?
clinically sterile or achieved by using like UV light and other forms of radiation, and people
Nope.
don't like the idea of their food being radiated?
That's possible, but you could. This equipment here, this went into an autoclave, just like we do for food. It went into an autoclave. So you could irradiate, but generally it's not. Your dentist office has an autoclave. They don't have a irradiating device.
Because of the temperature is too high, and it probably destroy the textures or taste of the food.
So exactly. So if you go back to my first lecture, when we talked about food preservation, what we're trying to do is preserve the food as best we can without changing, remember, the organoleptic, nutrition, all those properties of the food. So we could put this in an autoclave, or a retort is what we call it in food science, and retort the hell out of it, okay? So that it is clinically sterile. But now the peas and corn, we made pea soup and corn soup, okay? It's not peas and corns anymore. And we've destroyed the nutritional value of many of these foods. So why not? Okay, would it not be better if food were held to the same standard as pharmaceuticals? If they're not clinically sterile, why are they so well preserved and have such long shelf life? Okay, so in food science, we use this term called commercial sterility, not clinical sterility, commercial sterility. And the USDA, FSIS, has a definition. So if they use this definition for processing animal products. The condition achieved by application of heat, alone or in combination with other ingredients or treatments. So other ingredients could be nitrite, or they could be other salts and so forth, or low water activity, to render the product free of microorganisms capable of growing in the product at non-refrigerated conditions, kind of room temperature, at which the product is held during distribution and storage. And then the additional part of it, commercial sterility results in the products that are safe because the pathogens of concern are inactivated. The product is shelf stable as long as the container is intact, because any spoilage organism that could theoretically grow in the environmental conditions within the container, i.e. anaerobic, at normal storage temperatures, i.e. mesophilic, are killed. All right, so we got all that. Make sure you understand that. You don't have to memorize that, but make sure you understand that. So commercial sterility achieves a level of sterility that preserves the product while minimizing quality damage to the food. Here's the question. So what's left if it's, what's the difference between commercial sterility and clinical sterility? What's left theoretically after commercial sterilization? So if we go back, all these, going back here. Sorry. What's left? If you look at this definition, what could theoretically be left? Okay, what could be left? So those, that can of, those canned vegetables, the canned soup, or the aseptically packaged soup, what could be in there? Great question. So Rachel says spores. What kind of spores? Could CBOT be in there, Rachel? That wouldn't make it safe. It says the pathogens of concern are inactivated. So Rachel is half correct. Rachel, you want to amplify on that? Correct.
I'm not completely certain, but other spoilage organisms also make spores, don't they?
Yes, they do. Okay, so let's
So
go.
those could be in there.
Okay, so we say that the product is stable as long as the container is intact because spoilage organisms that could theoretically grow are killed. So if they're anaerobic and mesophilic, they're killed. So what would they have to be if they're not killed? Anaerobic and what could so when we put a food in a retort, and we're going to talk about the temperature in a moment, it's going to be, you know, way high temperature, 120 degrees centigrade, above boiling because we're going to actually put pressure in there. So what could survive that?
Thermophilic?
Aha. So they have to be thermophilic. So what's left are thermophilic or thermotolerant spores. Okay. But they're, again, if we go back, it says free of organisms capable at growing in the product at non-refrigerated condition during distribution. So they have to be, they're not capable of germinating in the food due to storage, temperature, pH, water activity. Okay. But we'll see in a minute, they could be. Okay, so yeah, we'll see in a minute how these can grow. So what's not present? Pathogens, vegetative cells. And so therefore commercially sterile foods are sterile except for conditions, or sterile except for spores that won't germinate under normal ordinary conditions. Okay, so the only way these thermophilic spores would germinate would be if the conditions were high storage conditions, like 55 centigrade, these guys could theoretically germinate. Okay, so let's make sure we're clear on this. Commercially sterile foods have no pathogens because all the pathogens that we know about are mesophilic, whether they're spores or not, so they're killed. And there will be no vegetative cells because they're definitely killed. Okay, so only thermophilic spores. But we don't worry about them because they won't germinate. Okay, everybody clear on this, hopefully? Okay, so Leslie on the chat put thermotolerant bacteria, but only the spores of thermotolerant bacteria would survive. Okay. All right, so now let's, again, come back to this notion of how we're going to set up thermal processing conditions. How do we know? how do we know how long to process? So if the goal of thermal processing is to kill microbes, but pasteurization or commercial sterilization, we have to be able to establish empirically the conditions necessary to achieve whatever that specific goal may be. So we don't try to guess, and we don't use trial and error. So we don't say, well, let's heat up that food to 80 degrees for 5 minutes and see if we killed enough. That's guessing, that's trial and error. We don't do that. Okay. We have to do this empirically. So that being the case, how do we figure out how long a given food should be heated and at what temperature? How do we do this empirically? Everybody understand what I'm asking? It's not trial and error. It's not like you bake a cake and you decide, should I put the cake in the oven at 325 Fahrenheit for 20 minutes to see how it comes out? And you could do that, and eventually you'll make a perfect cake, but that's not how we do it. We want to do it empirically. Okay, and so you can say, well, I can just look them up. They got to be published somewhere, and they sort of are. But we'll see that in a second. So conditions and parameters are not fixed for most foods, nor are they arbitrary, nor are they based on trial and error. Conditions are derived empirically and vary with the food and the level of destruction. Okay. And we want to do this without overheating. So in milk, they have been empirically derived. So for milk, you could actually look it up, but it was derived empirically. So for milk pasteurization, the conditions were 71.7, pretty precise to the decimal point, for 15 seconds. For egg pasteurization, so eggs can be pasteurized, liquid eggs, so you could crack an egg, basically scramble it up and pass it through a pasteurizer, and you could pasteurize liquid egg, and that's really common in the food industry. 60 degrees, 0.0 for 3.5 minutes. The roast beef that you buy at Arby's and at delis and at grocery stores, pre-cooked roast beef, again, 65 degrees for 1.5 minutes. When I say this time, that means it's held at those temperatures for that much time. Time and temperature. Fruit juice, that can vary, so I put some question marks there. And beer, again, depends on the manufacturer, that can vary. Okay. Now these foods are required by USDA. Some of these other foods are up to the, well, fruit juice has an FDA requirement, but beer would be up to the manufacturer. Okay, so how are we going to determine time and temperature for heat treating foods? Okay, so first we have to decide, what is the goal? Is our goal food safety? Is it to prevent spoilage and extend shelf life? Okay, and what are the relevant targets? Okay, so now this is why this is why this unit, I like to have, I think when I first started teaching this class a long time ago, we thought about maybe moving this unit up ahead in the semester, since it was so important, but we decided that it's best to put it at the second half of the class after you have some food microbiology knowledge, okay, regarding the troublemakers and the likelihood of their being present. Okay, in other words, you have to know what are the pathogens that we have to target. So you do know that by now. And so those would be things like Salmonella, Listeria, Clostridia, O157, even parasites. Okay, now we haven't gotten too much into spoilage microbes, but you know enough. We'll get to this eventually, but a bunch of spore formers, lactic acid bacteria, yeast, and mold. So we have to know who our targets are. Okay. So for example, in that pre-cooked roast beef, what's the target, you think? What's the target for pre-cooked roast beef? What's this 65 degrees for 1.5 minutes intended to kill? Not so much S-Tech. S-Tech would be perhaps, I'd have to double check, but I'm almost sure that this is set up for Salmonella. What about for egg? So this is Salmonella. S-Tech would be pretty close to it. What about egg pasteurization? What's the target? That's definitely Salmonella. because they're associated. So what I say is your food microbiology knowledge, you have to know a little bit about the ecology of those foods and what microbes are likely present, the ones that are caused, and they're going to cause the trouble. Okay. Okay, then the next question is, how many do we need to kill? So when we say the target is salmonella in pre-cooked roast beef, E. coli in beef, whatever the case, how many do we need to kill? That's a good question. How many is all? What is sufficient or required? How do we even measure cell killing? Okay, so let's continue. All right. Here is an example that I showed you before of growth of Lactococcus lactis at 32. Remember this one? We inoculate milk at 10 to the 5, and we do this incubate at its temperature optimum. And we have a lag phase, we have an exponential phase, we have a stationary phase, right? All right, let's say right at 13 hours, right there, we shift the temperature of our water bath quickly, or we just take the tubes out and we put them in a water bath at 70 degrees, 70. Okay, and now we're going to measure inactivation of Lactococcus at 70 degrees, and we plot those points. And we get this, these are our data points, and they can fit conveniently onto a nice line there. Okay, everybody see that? We grew them, and then we shifted them to a high temperature incubator, and that's what we got. Okay, so here is the little practice exam question. Which of the following describes the rate at which microbes grow and die? Grow linear, die linear. Grow linear, die logarithmic. Grow logarithmic, die linear. Grow at logarithmic and die at logarithmic. Okay, give me your answers. A, B, C, D. Everybody can answer that one. Looks like several of you like C. C. Hope you're just not following the trend. All right. Okay, Emily's hedging her bets a little bit. They grow logarithmically and they die logarithmically. Let's go back here and look. We'll show you this, but this is a log scale and this is a straight line. Okay, just like growth rate is logarithmic, straight line, killing is logarithmic, straight line. Okay, so microbes grow logarithmically and they die logarithmically. Implications of logarithmic killing. So in a given sample, can you ever kill all of the organisms in that sample? Can we get to zero? You give me a yes or a no. Okay, get some no's. Okay. All right, so there's my curve. We just showed that. Okay. Now, before we go too much further on this, let's review graph paper 101. Okay, here's some graph paper. Has anybody ever held a piece of graph paper in their hand? So when I was in college, you'd have to go to the bookstore and buy graph paper. You buy, 25 sheets for, a couple dollars. Okay. And that was before they even had, I guess they had copy machines, but copy machines wouldn't do a very good job of copying. And so you had to buy the graph paper. All right. So some of you have not seen graph paper. It's hilarious. Okay. Okay. But you know about graph paper, I think, from somewhere. So what kind of graph paper is this? This is just regular linear graph paper, linear on the Y, linear on the X. What kind of graph paper is this? Somebody just chime in. What kind of graph paper is this?
It's log graph paper.
It's log. Give me more specific information on
It's
what?
on the Y scale, it's logarithmic in terms of it jumps differently than the
This is
linear would.
log and this is linear. So we call this semi-log, right? Semi-log paper, okay? Because time is a linear function, but this is a log function, okay? And this is 3 cycle, 1, 2, 3 cycle. Okay. Some functions are linear functions. So your grade versus hours spent studying food microbiology is a linear function, right? So the more hours you study, the higher your grade. Okay. Here's a linear function, but it's the inverse function. Minutes spent on Facebook during my lecture, or whatever your social media you're on, is a inverse function. So it's still linear, but it's inverse. Okay, so here's my 3 log cycle semi-log paper. And if I were to call this 1, what's this point referred to here? What's this would be? This would be what? This line is, if I put the unit here, what would that be? If you could see it, pretty easy. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. What's this one up here? The line above 10. Please. This line is 20, 30, 40, 50, 100, right? Okay, what's this line down here? Where are we starting? Somebody tell me. Where's the graph start? 0.1, thank you. Okay. Okay, so that's 2 cycle log paper. So this was 3, this is 2. This is 1 cycle log paper. Okay, everybody follow that? All right. So let's go back. Microbes die logarithmically, they grow logarithmically, they die logarithmically. Can you get to 0? Okay, so now I have 1, 2, 3, 4, 5, 6 cycle log paper. Okay, and I'm measuring that same curve that we talked about before. So let me go back. Again, 10 to the 1 is 10, 20, 30, 40. 10 to the 2 is 100, 1,000, 10,000. And that's 10 to the 0, that's 1. That's 10 to the minus 1, that's 0.1. And that's 10 to the minus 2, right? 0.01. Okay, right? And so if we plotted the survival of L. lactis at 70, and these are, I can only get a few data points because once I get below 100 cells per CFU, I don't have any way to measure those really, right? But I could extrapolate that line. And if I went all the way out to 12 seconds, I have, how many bacteria do I have per mil? I have 0.1. Remember we said this was 0.1. I have 0.1 CFU per mil. So can I have 1/10 of a cell? That doesn't make sense. So have I killed all of the organisms in that sample of milk, in that milliliter? Have I killed all of the microbes in that, let's say it's a bottle of milk? If I'm down to less than 1 cell per milliliter, have I killed them all? Give me a yes or a no. Okay. Important concepts. No, the answer is no. Okay. Based on the data from the preceding data, after 12 seconds of heating, the milk contains, I already gave you the answer. No viable bacteria, no. A non-measurable number of bacteria, less than 1 cell per mil, none of the above. The answer is less than 1 cell per mil. Okay, so let's look at this again. Here is my beaker of milk. It has 10 CFUs per mil. So if I drew off a milliliter out of this flask of milk and I got lucky, and the milk was very homogeneous in that mill that I then put on a plate, I would get 10 colonies because I have 10 CFUs per mil. If that milk contained 1 CFU per mil and I pulled the mill out and I got lucky because I could have pulled a mill from a space that didn't have any cells, but if I got lucky and got a perfectly representative sample, I would get 1 colony on that plate. But here I have a mill out, the chances are I'm not going to get a colony on that plate. If I took 1 mil, chances are 10 to 1, I won't get a colony on the plate. But if I took a 10 mil sample, I would probably get, and I could spread that 10 mil sample on, I can't really do it very well on 1 plate, but I could spread that on a whole bunch of plates. And one of those plates, I will get a colony. So that has 1 CFUs per 10 mils. Everybody see all that? You know, going through kind of pretty fundamental microbiology, but I wanted to make the point. Okay, that's the same as 0.1 CFU per mil. So what we can't really ever get to 0 because in fact on log paper there is no 0. It's always going to be, if I took 20 cycle log paper, it's going to be 0.0000001 at the bottom. There's no 0 in log scale. So what we're really asking is, what percent should be killed? 50, 90, 99, 99.9. That's what we're really asking. Again, so we need to rely on our food microbiology knowledge to know how many microbes are likely to occur in that food, or at least what are the risks we wish to take. Okay, and so we keep score using log scales, okay, because they die logarithmic, so it's easy to measure killing based on log reductions. So we may want to say we're going to try to kill 1 log, 3 logs, 5 logs, even 12 logs. Okay. So then all we to actually convert a log into time, all we have to know is how much time it takes to kill 1 log. And then we could say 5 logs would be 5 times that. Okay, everybody with me? Okay. And the log reduction is determined experimentally. Remember I said we're going to do this empirically, experimentally, and that's exactly what we're doing here. We're going to determine the log reduction experimentally via what we call survival experiments. And in your problem set, I gave you survival data from a survival experiment. And this is simply done by preparing tubes or containers. They could be sealed tubes, they could be cans. We inoculate them with a known number of microbes or spores, incubate them at a constant temperature, whatever the killing temperature that we choose, and then we remove, immediately cool, and enumerate. And then we plot survivors on a log scale versus time on semi-log paper. So for example, we would take a sample and we would put, in this case, I put 0.1 mil in each of these tubes, put them in a water bath and incubate, and then we take them samples out and plate them. Okay, and we would call this a survival curve or a rate of destruction curve. I like survival curve. And we plot on the y-axis survivors. I've labeled this as CFUs per mil. And when you do your problem set, make absolutely sure you label the title of the figure and the axes and use the right units, 10 to the 7, 6, so forth. And on the x-axis, we're measuring time in seconds. This happens to be 80 degrees. Did I say that anywhere? Yeah, 80 degrees. Okay, and we plot this data. Now, don't forget, it's below some point. We don't have data points because it's hard to enumerate once you get below, well, 10 to the 3 or so. Okay, but in any event, we can make a straight line from this data. And when you do your problem set, you can use Excel if you choose, and that will give you the straight line. You could use your hand calculator to give you a straight line. For the purpose of the problem set, I am fine with an eyeball method. Okay, do your best to make a straight line. Don't connect the data points because that will give you a crooked line. You make a best line, a regression line. Again, use your hand calculator, Excel, or your eyeball to make a best line. Best line is simply what your calculator is doing is giving you a line where it makes the data points closest to the line as possible. You can do that by eyeball. Okay, so you make your line, and now you've to determine a log reduction, you simply see where this line crosses. In this case, I use 10 to the 6 and 10 to the 5, or you could use 10 to the 5 and 10 to the 4, but you want to get 2 points that are on the line. This is a log reduction, right? From here to here. How long did it take to make this log reduction? Easy. We just read down. So at 10 to the, at 1.5 minutes, there were 10 to the 6 microbes in that still surviving. And at 4.25 minutes, there were 10 to the 5 microorganisms surviving. So this is a log reduction. And how long did it take? Okay, we're going to show that in a second. But that time at a given temperature, that was, what do we say, 80 degrees, to reduce the population by 1 log is called the D value. It's one of the most important terms in food microbiology, the D value, the decimal reduction value, the time at a given temperature to reduce the population by 1 log. Okay. And you could actually calculate it from the slope. Okay, you can calculate from the slope where it's 1 minus the, where it's the reciprocal of the negative slope of the survival curve line if you wanted to make a regression line out of that. Okay. A 1 log reduction is the same as a 90% reduction. If you go from 1,000 to 100, that's a 90% reduction. That's exactly what we're doing. 2 log would be 99%, 3 log 99%. So that's why you could say, that's why you see things all the time like sanitizers will, mouthwash will kill 99% of the microbes in your mouth. That means you got a 2 log reduction. Okay. Now, if we go back to our survival curve, here we go. That time from here to here is 2.8 seconds. Therefore, the D at 80 degrees is 2.8 seconds. So for your problem set, you're going to have to figure out the D values. You can put them all on one curve, which is how I would suggest you do it. And you calculate the D at every single temperature. All you need, I think, here I show you 1, 2, 3, 4, 5, 6, 7 data points to make a line. But in theory, how many data points do you need to make a straight line? In theory, you only need 2, right? 2 points to make a straight line. You have to have at least 2. More is better because then you have more confidence in your line. Okay, but that's what it takes. The D value is 2.8 seconds. Okay, now we'll finish up in a minute. I want to get these last important concepts noted. D values relate to the resistance or sensitivity of that organism to heat. The higher the D, right, the longer it takes. Instead of 2.8, let's say it took 5 seconds or 10 seconds to kill 1 log. The higher the D, the more resistant is that organism to heat. D values depend on the conditions of the survival curve experiment, for example, pH, medium constituents, so forth. We'll come back to that. So for example, CBOT at 121 has a D at pH 7 at 0.21 minutes. But if we acidify that food to 5, it makes them more sensitive. Remember, higher the D, more resistant, lower the D, more sensitive. Okay, last slide. I'll leave you with this thought. A student does an experiment. They have 3 cultures: Strep thermophilus, Listeria monocytogenes, and Pseudomonas fragi. They do survival curves for all 3 of them, but the labels fell off the tubes. So all we know is we have 3 survival curves. Which is which? 3 survival curves. Which is which? Which is A? Somebody tell me what A is. This is a really good question. Should I leave this for you and you can ponder it? Let's answer it now. Streptococcus thermophilus, Listeria, Pseudomonas. Which one is A? Leslie, why'd you say A? Why'd you say that was Streptococcus?
Because the survival curve is not really that steep compared to the other lines. So and from the word and from the name Thermophilus, it means it's somehow like
Good thing.
thermotolerant.
Okay, so Leslie's right. The name tells you it's thermophilic and has a curve, a death curve that has a higher D value. So I'm not going to figure out what this is, but it has a higher D value. And she also knows, well, what's the next one? What's C?
Listeria.
Is what?
Listeria.
Okay,
no.
somebody else want to offer up an answer?
It's Pseudomonas because they are psychotrophic, are sensitive to heat.
Antar's correct. So Pseudomonas, we know grows at low temperature, and then we said it's really sensitive to heat. So this D value is really small. Okay, and it's by process of elimination that we would say Listeria is B. Okay. All right, so we'll stop there, and we're going to continue this discussion on Wednesday. I really advise you look at the problem set, look at these notes that I'll post, look in the book if you have it, but really know this stuff well because it's going to be really important concepts. Okay. Sorry for keeping you an extra few minutes. See you on Wednesday.